Well, thanks, Martin. Thanks also, Beatrice and Debbie and Luis. It's such an amazing conference. I'm super delighted to be part of it. So thank you for inviting me. So my life bio, we have a disclaimer that you should all be aware of. There will be some forward-looking statements, but those forward-looking statements are going to be about three things. So the first is I'm going to talk about why longevity science is such a good bet. A good bet if you're thinking about it as a career, if you're thinking about it as an investor, as individuals, and for society. And then second, I'm going to talk about we've actually been stagnant for a while, and we need a breakthrough. And the third point is that I think I know what the breakthrough is, and I'm going to try to convince you of that as well. I certainly voted with my feet on it. So a good starting point on that first element is this curve. This is a survival curve, also called a Kaplan-Meier curve in humans. Starts at 100%, goes down to 0 at increasing ages. And you can see it's plotted here for 4 different cohorts. And over the last 150 years, there's been great improvement in the shape of that curve, largely due to sanitation, vaccination, and reduced infant mortality. And so we're actually pretty good on the shape of the curve, but there are 2 big things missing. The first is health span. It's the quality of those years. Are you spending it sick with frailty or some other disease? And then, of course, we haven't increased lifespan despite the improvements we've made so far. Now, these 2 things are actually linked. And the way to see that is this chart, which shows that the number 1 risk factor for the disease, most diseases that afflict us, actually more than 90% of our mortality in the developed world, are diseases of aging. So you can see here plotted cardiovascular disease, cancer, dementia, diabetes, really the scourges of mankind. They're all going up with age and exponentially so. And there, from Linda Partridge's lab, you will find there are at least 300 others that I could have put on the plot here. So if we can manipulate the rate of aging, slow it down, reverse it potentially, then not only can you address lifespan, but you address health span as well, because you address the incidence of disease and frailty. Is there a reason to believe that that is possible? There's actually a lot of evidence. First line of evidence comes from genetic variants. This is in worms. Again, we're looking at a survival curve or a Kaplan-Meier curve. The white dots are normal worms. The black dots are worms with a single mutation in a gene called DAF2. You can see they live twice as long. They're also healthier along the way, and it's a gene involved in nutrient sensing, caloric restriction, diapause, things like that. Second, data from pharmacological interventions. This is an example from mice. Again, we're looking at a survival curve. The black line are normal mice. The red are mice given just one drug called rapamycin that works on a target mTOR, which is downstream of insulin signaling, again, associated with this shifting a state of energy from maintenance and repair to growth and reproduction. And then third is evidence from epidemiology. This is in humans. A different chart here. Now we're looking at lifespan versus caloric intake across different populations of Japanese, sumo wrestlers, mainland Japanese, and Okinawans. And you can see there's an inverse correlation between the amount of caloric intake and lifespan. Pretty much all the blue zones, Okinawan's probably the most famous blue zones, all the blue zones have some level of caloric restriction. So lots of reason to believe that lifespan is manipulable and health span along with it. There's also a lot of reason to believe that it's super valuable. Now, many of us, hopefully most of us in this room, kind of fundamentally believe that at a moral level. Life and health have value. That's the end of the story. That's all you need to say. However, there are plenty of other arguments in support of that if you don't accept that piece, which are there are other benefits, including tremendous financial ones. So a lot of serious economists have looked at this and have concluded there's trillions of dollars of value. And it's through 3 things. It's through more addition to GDP. So if you're healthier longer, if you're living longer, you can work longer if you want. You are a consumer longer, which drives the economy. The second is less reduction from GDP. So again, healthier longer, living longer, you're delaying and shortening the period in which you need to draw down entitlements, both healthcare and otherwise. And then third, these economists put in a value of life itself, which you might say is infinite, but they calculate a value to it. Okay, so I've hopefully convinced you of point one. Probably if you're in this room already, you believe it. But there's a lot of reason to bet in this area because there's evidence that it's highly feasible and there's evidence that it's highly valuable. However, we have a problem, which is we've been stagnant for a while. A way to see this is to look at the best known lifestyle intervention, caloric restriction, known really to some degree since antiquity, but studied scientifically from the teens, 20s, and 30s. And the best evidence for a therapeutic intervention in mammals is rapamycin. It was the first one that was shown to have an effect in the National Institutes of Aging Interventions Testing Program, and it's still the best. This is a Lee paper is actually from Matt Kaberlein's group, who's here in the audience somewhere. Another way to see that is just looking at modern advice versus historical advice. There was a great paper done at the Veterans Affairs in the US where they looked at 8 things that together can extend your lifespan or shorten your lifespan by 24 years. Kind of things we all know, right? Exercise, diet, sleep, don't drink, don't smoke, don't do drugs, manage your stress, manage your social relationships. But if you go back to Sir Francis Bacon, every single one of these is in his writings. The language is sometimes a little bit colorful, like poppy juice will make the spirit curdle, or tobacco doth like poppy buzzle and trouble, but I'm assuming those are bad things. But they're all there, including some current fads, like total plasma exchange. He had an opinion on, and he thought it was unlikely to work, that in old age, sucking the blood out of the arm of a young man is a vain imagination. So we need to improve. And some of the things that we have on the table right now, the best current interventions that I just showed you are unlikely to change lifespan and health span in humans in the same way that they change it in some of these other organisms. There's a reason theoretically to believe that things like caloric restriction will have a much more profound effect on short-lived organisms than on long-lived organisms. And there's a reason empirically to believe that as well. If you just plot the percent lifespan extension versus the underlying lifespan of the organism, and there's a little bit of noise around these dots. I've picked specific papers quoting a lifespan extension. You can find papers with different numbers, but roughly speaking, you get this inverse correlation down to the point of rhesus monkeys, which I've plotted here at a 6% increase from caloric restriction. There were 2 papers on rhesus monkeys, quite famously. One said 6%, the other said 0, because it's really getting down at noise level where you probably can't find it, and always going to detect it. But if you extrapolate this line to humans, you get a 2-year life extension from caloric restriction, which is not nothing. I'm not saying it's a bad thing to do because it should improve health span along the way and should increase your chances of reaching a ripe old age. But it's not going to fundamentally change lifespan in humans. And another way to see it is the same epidemiology data that I already showed you across populations of Japanese. If you just extend that line to basically the maximum, you get about a 2-year impact. So we need something more profound than the sort of best known state of the art right now. And I actually think we've finally sussed it out. The insight starts with this. So everybody in this room, believe it or not, started as a single cell. You were once one cell, and that cell divided and divided again. And as those cells divided, they gained identity. They went down different lineages. Some became nerve cells, muscle cells, skin cells, and so on. That's during development. And then as we age, those cells gradually lose identity, and that is associated with a loss of function and increase in cancer. Built within us, that completely reverses this, because when we reproduce, our old cells combine to create a baby that has 0 days of age. And that is incredibly everyday, and we all know it, and it's incredibly profound. There's biology in us that resets the aging clock. There has to be, otherwise the germline doesn't, persist. And Shin Yamanaka tapped into that biology and won the Nobel Prize in 2012 by creating what's called induced pluripotent stem cells through the process called epigenetic reprogramming, applying just 4 proteins, abbreviated OSKM, that can take any cell and bring it back to pluripotency. Now, people focused on the fact that these cells were pluripotent. They could then, they had no identity. They could then redifferentiate into anything. But hidden in his discovery was the fact that the cells were completely de-aged. So when you take a cell and you induce this pluripotency and then you grow a clone from it, that clone starts as a baby. And it's now been shown you can take even the cell from a centenarian and completely de-age it this way. You can also do it over and over again, at least 25 times in series in mice. We don't clone humans. But you can take a cell from a mouse, clone it through this process, starts again as a baby, grow that clone to an adulthood, take another cell from that, reclone it, starts again as a baby. So the thing that we find conceptually hard to believe, that we can de-age cells, we already do. It's routine, existing technology. We know how to de-age your cells. The problem is, you can't do that in vivo because you need in your body because you need your cells to retain their identity. Otherwise, you cease to function. If your nerve cells are no longer nerve cells, your muscle cells are no longer muscle cells, that's a problem. There's also a risk of cancer. But the insight that came out of David Sinclair's lab at Harvard that was on the cover of Nature in 2020 is that if you treat with just 3 of these 4 factors, abbreviated OSK, what they saw in the paper was that there was no pluripotency. They did not see NANOG, which is a key marker of pluripotency. They did not see tumor formation. But they did see rejuvenation effects at the cellular, tissue, and organism level. And so the company that I joined, before I do that, I'll just, one other way to kind of see this is, so the central dogma is that DNA is transcribed to RNA, which is translated to proteins, which then catalyzes the metabolites that are active in your cells. You can ask, what is different across 2 different cell types, say a muscle versus a nerve cell, or 2 different cell ages, an old versus a young cell, and work backwards? Well, they have different metabolites. I can actually tell you, just by the metabolite profile, what kind of cell it is you're looking at, old or young, nerve or muscle. They have different proteomes, they have different transcriptomes, but they have the same DNA. And it doesn't take a rocket scientist to ask the question, well, what is it that's happening here that could be the causal step? And that is epigenetics. And so if you can do this partial epigenetic reprogramming, not taking it all the way back to stemness, but partway back, you can potentially de-age. And so the platform that my company is working on is this platform of OSK. We've licensed the IP out of Harvard. There's very strong IP around it. That has a lot of different applications, right? Every age-related disease, potentially aging itself. But of course, to reduce that to practice in a pharmaceutical setting, you want to go after specific diseases and start someplace. And so we're starting with optic neuropathies. Many people have heard of glaucoma, which is one of the optic neuropathies. It's actually the leading cause of blindness in people over 60. We're also going after NION, non-arteritic anterior ischemic optic neuropathy, which most people have not heard of, but it's the actually leading cause of acute blindness in people over 50, and it has no treatment. We're going after it with a treatment called ER100. It's an inducible system, so it induces the expression of OSK in cells only in the presence, only in the presence of doxycycline. So if you look on the right, the top picture is in the absence of doxycycline. There's no dots, so the dots are expression of, in this case, K. And then in the bottom, you can see in the presence of doxycycline, it's on, it's being expressed. So that means we can turn it off, which is great for a first product on market of this kind because we want to make sure it's safe. We always have the fail-safe of being able to turn it off if we want to. In the original paper, they showed efficacy in multiple mouse models. The first is crush, like literally physically crushing the optic nerve. You can see in the picture there, after the crush, there's no axon signaling down the optic nerve. And then in the bottom picture on the left, after treatment, the axons are literally regrowing back down the optic nerve. They grow all the way back to the brain. Second, in a model of glaucoma, where they recovered vision all the way back to baseline. And third, a model of aging, again, vision recovered all the way back to baseline. And vision declines with age, obviously. They also, as I mentioned, found safety with no nanog, no tumors, and this is over an extended period of time. For the treatment, what you saw on the prior page, you only need to dose for a month or two, but in the safety experiments, they actually dosed for most of the lifespan of the mouse, and again, saw no serious problems. We've since, at Life, recapitulated a lot of the work that came out of the lab there in non-human primates, so NHPs, and really the full logic chain now exists across those two species that you would want to see in preclinical data. We show that we get delivery. In the second column, we show that the methylation state, the epigenetic state, changes exactly back to baseline. The third, fourth, and fifth, we show that there are improvements at the cellular, tissue, and organism level. And then, as I mentioned, safety. And I'm happy to report that in our GLP tox, which is required for FDA filing, we had no serious adverse events in the non-human primates. So we intend to go into clinic at the beginning of next year, so just a few months away now. Our understanding is this will be the first partial epigenetic reprogramming therapy in clinical trials. It's a phase 1 study, so phase 1 studies, as you know, are designed to test tolerability and safety, but we will be dosing patients, and so both in glaucoma and NION, and so we should get at least some anecdotal data around efficacy. So I hope I left you with just 3 points. So this is a great bet because there's a lot of evidence on feasibility, a lot of evidence on value, that we really need a breakthrough, and that I think this particular platform is one that has the chance to be that breakthrough. I do think there's value in multiple approaches, so I'm glad that as a group we're trying to tackle this problem through multiple angles, at one way or another, deliver what's on the page here, which is extension to personal health, both lifespan and health span, huge economic value, and I believe changing the world in a way more profound than the examples that are shown on the page here, like flight, computers, the internet, all of which at one point seemed absolutely impossible to people, inconceivable, and yet now we take absolutely for granted, and I believe that's how we're going to feel about aging technology in the near term. So thank you.
Great presentation.
Thanks, Matt.
Despite what people think, I'm actually pretty bullish on this technology. I do want to ask you, though, about your feeling as to whether this is really the breakthrough or a breakthrough. So you showed, some previous lifespan effects. Rapamycin, you can get to about 30% in mice. Weindrich and Walford got 60% in mice with caloric restriction. The best people have done, to my knowledge, with reprogramming is less than rapamycin for lifespan, right? And public comments by Hal Barron suggest multiple rounds at Altos haven't done better than rapamycin. So what do you think about that? What does that mean? Do you think this really will be the path to get past those barriers?
Yeah, so it's a great question. It was one of the ones that was on my mind as I was, deciding did I want to bet on this. There's some curious results, which I'm sure you've seen out of, I think it was Retro, out of George Church's lab, where they tried it in very old mice and got 109% extension. It's sort of it's hard to compare that to the other examples.
It's not that hard.
Right. There's only one study that I know of that showed a lifespan extension from epigenetic reprogramming or partial epigenetic reprogramming in mice. And it's not as profound as some of the numbers that you just mentioned. Do I think we've got the exact right formula, the exact right formulation and approach to it? No, not yet. I think we have enough of a starting point. The reason why I'm bullish on it isn't, I mean, there's empirical data already, which is great, but it's also just the fundamentals of when thinking about the way that aging is programmed in, which you have to buy into a belief that I have of aging is an evolved trait. It's been selected for. There are organisms that don't age, like many bacteria, and there's a reason why aging has evolved in it, and it's a whole nother talk, which I've given elsewhere. And that in order for that to be controlled, there's a mechanism that must exist to do it. And that mechanism has to have a reversibility aspect to it, which allows you to reset it when you reproduce. And there's a good reason to believe that we've seen it now. Like, it's the same thing that resets cell identity. By the way, this was presaged in the evolutionary literature back in the 70s, evolutionary biology literature. In 1975 and 1984, there were two papers by Robin Holliday, who was the advisor for Tom Kirkwood, saying, aging is quasi-programmed, it's not directly programmed, but there's a reason why it's evolved. And I think the way it's managed is through DNA methylation. Incredibly prescient, 1975. Do we have the exact right technology right now to manipulate it? I don't think so yet. I think we've got a great starting point that can help people in real need. But I think fundamentally, I believe enough of the story that this is the thing that's actually the causal factor to say this is where I want to focus my energy.
First of all, great presentation, as usual. And what got me involved into longevity was Yamanaka paper in 2006. I thought this was magical, absolutely magical. This was life-changing. But a few days ago, you probably know, Shift Biosciences, Daniel Ives, he announced that you don't need the Yamanaka factors. You need only one protein. So if this is true, this is even more magical. So what is your take on magic?
So I'm really happy that this field is tremendously underinvested in. Like we have to remember just holistically, like I care about advancement in this field, and numbers vary on it, but there's something like 400 million is the budget for the Division of Aging Biology within the NIH versus a $50 billion total budget, so 1%-ish. There's only a couple hundred companies in this space versus 5,000 companies working on oncology. I really want there to be multiple avenues of approach. So I'm really happy Shift is doing what they're doing. There are some other great companies focusing very much in our area, like Altos and New Limit, and I'm really happy they're active. And I think through the collective efforts, we'll find multiple things. There are lots of diseases to solve. So there's plenty of room for multiple companies here.